My name is Muhammad Shazwan bin Muhammad Alwi, and this is my group members.
Hi, Assalamualaikum. My name is Muhammad Azawdi Mirza Abba. Okay, hi, good morning. My name is Luis Razul.
Hi, good morning. My name is Muhammad Faris Daniel.
Hi, good morning. My name is Sharil bin Matsa. Practical 1, Spectrophotometry, Protein Analysis.
So today we're going to demonstrate the use of spectrophotometry for qualitative and quantitative analysis of biological samples and molecules.
Objective: to determine the absorbance spectrum of a protein sample using the Bradford assay. Number 2, to calculate the protein concentration in unknown sample with the equation obtained from the standard calibration curve. Number 3, to assess the efficiency of the Bradford test for protein analysis by examining the level of consistency of duplicate reading and the behavior of the standard curve. Number 4, to construct and evaluate the standard calibration curve using existing amount of BSA protein to estimate unknown protein values. Number 5, to statistically analyze experimental data involving the calculation of mean, standard deviation, and error bar, and to comprehend their significance in scientific measure. In this spectrophotometry experiment, we learned how to measure the concentration of protein using a spectrophotometry and the Bradford assay. By preparing a standard curve with non-protein concentration, we were able to determine the concentration of unknown sample based on their absorbance readings. The Beer-Lambert law helped us understand the relationship between absorbance and concentration. Although they were challenged with a sample preparation and ensuring accurate reading, we gained important skill in using laboratory equipment, handling sample, and analyzing data.
I'm Luis. In this experiment, I learned how the Bradford reagent is used to measure protein concentration based on absorbance. It really helped me understand how absorbance relate to protein level, which help us to calculate unknown sample concentration. One challenge I faced during lab was pipetting accurately, and timing the spectrophotometer reading was also tricky, as the color changed quickly. So to improve, I would focus more on precise and better timing when taking reading. Thank you.
Assalamualaikum. I am Muhammad Shazwan bin Muhammad Alwi. In this lab, we have learned how to use the spectrophotometry to measure protein concentration using the Bradford assay, and then we have learned the essential lab skills such as how to use pipette data analysis and interpreting the result using the theoretical concept, which is we use the true equation, which is y equal to mx plus c, and then the overall It taught us the importance of careful handling, clean the equipment, and then the good lab technique. So that's all from me. Thank you.
So my name is Shahri Matsa. So for our reflection, I learned how a spectrophotometer works to measure light absorbance. Other than that, I can learn how to prepare standard curve to estimate unknown concentration. And last one, I can learn how to use Bradford reagent to detect protein levels. The challenge that I face is handling the cuvette carefully to avoid a scratch, fingerprints or bubble that could affect the reading. Other than that, to keeping the accurate records of sample levels to avoid confusion between different samples. My suggestion to improve this practical is to clean the cuvette carefully and check for bubbles before measuring absorbance. Other than that, label the samples and cuvette clearly and use a sample tracking sheet to avoid mix-ups. Other than that, repeat the measurement to improve accuracy and make sure the results are reliable.
After conducting the experiments, the new skills that I learned from the experiment is how to use the micropipettes in a proper way. And the challenge that we face is the protein reading from the spectrophotometer. We need to do 2 trials to get the accurate result. And what we can improve from the experiment is if the lecturer has uploaded maybe a video or tutorials for the experiments that we are going to conduct, we need to watch it first so that we are not doing any mistakes or repeating the same mistakes as others.
First, one of the new things we learned was how to properly use a spectrophotometer and prepare a standard curve using Bradford assay. It really helped us understand how to measure protein concentration accurately. Second, one of the main challenges we faced was getting inconsistent absorbance readings, especially for the 100 microliter and 200 microliter samples. We had to repeat those parts because the results didn't follow the expected trends. Lastly, for improvements, we think more emphasis should be given to proper pipetting technique and handling cuvettes carefully. That would reduce errors and save time during the experiments.